Hello guys, today my topic is physics of protein. The goals of protein physics. Proteins are invariable participants in all life processes. Protein enzymes catalyze all chemical, electrochemical, and mechanical processes taking place in cell and in organism. The most important function of proteins is that of enzymes. Specialized enzymes serves as catalyst for all metabolic reactions, DNA replication, the transcription of the genetic message from a DNA strand to mRNA, and the transfer of the information encoded in the mRNA to the ultimate structure of protein molecule. Protein also function as regulators of the genetic functions of nucleic acid. Regulatory enzymes, also called allosteric enzymes, the movement of cells, organisms, the performance of mechanical work by them is realized by special contractile proteins, which serve as working substance for these processes. Contractile proteins perform the enzymic ATPase functions, accomplish the conversion of the chemical energy to mechanical energy. The charging up of the storage battery, like oxidative phosphorylation, occurs in mitochondrial membrane, and the requisite invariable participation of the respiratory chain enzyme. Oxidation reduction enzymic processes also occur in photosynthesis. Outer membrane proteins are responsible for the active transport of molecules and ions through the membranes, and consequently for the generation and transmission of the nervous impulse. Proteins control all metabolic and biogenetic processes. In animals, organisms, there are special proteins which perform specific functions. The protein serves to store oxygen and transport it. Low molecular weight proteins, or more exactly polypeptides, are hormone. The gamma globulins of higher organisms protect them against foreign biopolymers by functioning as antibodies in immune process. Proteins that enter into the composition of connective tissue, cartilage, and tendons, and also the proteins of skin and hair and feathers are structural proteins which provide a reliable and mobile active intercommunication between the organs, the unity of the organism, and its protection against internal influences. One can say that proteins are invariably involved in the storage, transfer, transformation, recording, and reception of chemical signals. Macromolecules, molecules, and ions in living systems. In many cases, the proteins themselves serve as signals, their receptors and transformers. Proteins that enter into the composition of the receptor systems of the organism records external signals into the chemical and electrochemical language. Protein molecules are the most complex of the molecules known to science. Their biologically functional spiral structure and also to the structure of supermolecular systems containing proteins are determined both by chemical bonds in proteins chain and by the entire set of weak interactions. Native proteins are never down coils. Globular proteins are aperiodic crystals of complex structure. They are dynamic rather than static systems, a sort of machine whose behavior depends on the position and properties of all its component parts. Apart from globular proteins, there are also exist fibrous proteins, contractile, and structure proteins. Protein denaturation. Protein denaturation is the loss of biologically functionality by a protein when it is heated, acted on acids and bases, etc. Consists in the destructions of weak interactions and as a result in the final transition of the aperiodic crystals into a random coil. Let us formulate the principal goals of physics of the protein. First, the theoretical and experimental investigation of structure of proteins, molecules, and supermolecule systems that contain such proteins. Second, directly related to these investigations is the problem of establishing the relationship between the primary structure of the protein chain and three-dimensional structure of native macromolecule. Third is the study of physical mechanism of formation of the primary structure of a protein, like the mechanism of genetic coding, translation of genetic information, and the regulation of the actions of genes by proteins. The study of physical mechanisms that control the various biological functions of proteins, primarily the enzyme activity. The solution of biological phenomena in cells and organisms on the molecular physical basis. And now we will talk about conformations of the polypeptide chain. To understand the protein structure, it is necessary to consider the possible conformations of the polypeptide chain. The conformations are primarily determined by the structure of peptide bond. The fully extended chain, measure of the valence angles, and change of the bond lengths has a trans conformations with zero values of the rotational angles. Such a conformation is not, however, the most stable. The hydrogen atoms of the imine group N and H form hydrogen bonds with the oxygen atoms of the carbonyl groups. In order to find the most stable conformations of the polypeptide chain, it is necessary to minimize its total energy, including the energy of the intramolecular hydrogen bonds. Thus, the hydrogen bond stabilizes the fixed conformations of the polypeptide chain in solution. The fixed conformations is commonly referred to as the secondary structure of the chain. The presence of the secondary structure, which is periodic, means that the chain is similar to a crystal. Let us now consider the relationship between the energy of the polypeptide chain and the angles of internal rotation, called steric contour maps, which resemble geodesic contour maps. The conformational energy of the chain is determined by the weak interactions of non-bonded atoms. Because of the planar structure of the peptide group, the rotational angles vary within the limits allowed by the overlap of the atoms of the peptide group linked by. Then there is not such a combination of these four angles at which the steric interaction of the unit is possible. Therefore, the polypeptide chain has a limited cooperativity, the range interactions in it being restricted to nearest neighbor interactions. This allows one to deal separately with conformational energies for individual amino acids. Residues, the steric map of for a particular residue depends subsequently on the nature of its radical. It may be presumed that the interactions in a given pair of peptide groups characterize the amino acid residue that links these groups together. Ramchandran studied the dipeptide glycine L-alanine and obtained a conformation steric map of alanine. The calculation was based on assumptions that atoms are rigid sphere having van der Waal radii determined from data on internuclear distance in molecular crystals. Using the potential of rigid spheres, one can efficiently obtain only the allowed and forbidden regions of values. Thank you, that's all.